Right now, two pieces of your brain are sitting outside your skull, exposed to the open air, protected by nothing more than a thin layer of tissue and fluid. They're your eyes. This isn't a metaphor. Embryologically, your eyes are outgrowths of the diencephalon, the same region of the developing brain that becomes the thalamus and hypothalamus. Around day 22 of embryonic development, two small pouches called optic vesicles bulge outward from the neural tube. They push through developing tissue, reach the surface ectoderm, and induce the skin to form a lens. Your retina, the light-sensitive layer at the back of your eye, is made of genuine brain tissue. It contains 5 types of neurons, including photoreceptors, bipolar cells, and ganglion cells, arranged in layers exactly like cerebral cortex. When light enters your eye, it passes through the entire thickness of the retina before hitting the photoreceptors at the back. These rods and cones convert photons into electrical signals through a process called phototransduction, using a molecule called retinol that physically changes shape when struck by light. This shape change triggers a cascade of enzymatic reactions that generate a nerve impulse. A single photon can trigger a detectable response in a rod cell. Your retina processes roughly 10 million bits of visual data per second. But it doesn't just relay raw data, it performs its own pre-processing. Edge detection, motion detection, contrast enhancement all happen in the retina before a single signal reaches the brain via the optic nerve. The optic nerve itself isn't technically a peripheral nerve. It's a tract of the central nervous system, wrapped in meninges and bathed in cerebrospinal fluid, exactly like the tissue inside your skull. This is why damage to the optic nerve doesn't regenerate the way other nerves can. It follows the rules of brain tissue, not peripheral nerve tissue. Your eyes even have their own immune privilege, a special status that suppresses inflammatory immune responses to protect these fragile outposts of the central nervous system. And right now, these two extensions of your brain are collecting 120 million data points of light and feeding them directly into your visual cortex.